So is that coming through for everybody?
Yes, looks good.
Okay. Thank you very much. So we have a couple of things to look at today. One is, of course, what my research group is doing, and a little bit about that. And Matthew Walsh is involved in that. And I don't want to steal any of Matthew's thunder because it's, it's pretty cool. So I'm going to kind of just introduce what we're doing at Swinburne, a bit of my background as well. And then we've got a video presentation to show about another project that Sonification is becoming part of. Now let's see if I can. Yep. So my background is that of a computer musician. So I began in the late 70s with my first synthesizer that I purchased, and continued from there. And it's a bit of a challenge to describe my instruments to blind people, but they are really stunningly beautiful leather objects that have integrated sensor systems, and they don't make sounds usually of their own, although some of them are hybrid instruments and will use an acoustic sound and signal processing to create music. So if you've never seen any of the instruments of the bent leather band before, it's a bit like something out of a Dr. Seuss book, crossed with a very nice fine pair of leather shoes. And in fact, I'm actually looking at leather shoe museums in Europe to be potential exhibitors of some of the work. A lot of this leather work was done with a Tasmanian artist who's a really famous leather artist. But I've also made instruments in fiberglass and all sorts of things, some studied musical instruments, activity theory, all sorts of systems. My background is kind of a musician, but I've moved into design. And in more recent years, I've moved from audio signal processing to video signal processing, and I've been teaching that at Swinburne University in the design school for many years. And as part of that work, been doing data visualizations, which of course is of some interest, but of course data art or sci art or data painting, or there's a whole range of terms, is a lot more fun and interesting. So I've been doing a lot of that. But at Swinburne, I had a chance to get back into audio. So with another colleague, David Sly from Clinical Technologies, we formed a research group called the Swinburne Sonic Group. And we have, we really enjoy that group because we have these amazing specialists that are in that group, including Matt Walsh and other incredible coders and people who are doing exhibition work in science and STEM, or STEAM. And there are artists there that work with the Osgrav people doing their outreach. And there's the wonderful Erica Tandoori, who's right here today from the Monash Biomedicine Discovery Institute. So what do we do at Swinburne? We do spatial sound, and really interested in that. That's an extension of my computer music work because a computer musical instrument is not an acoustic instrument. It can do other things. It can locate the loudspeaker away from the instrument. And what does that mean? That means that you can image sound in 3D. So I'm really interested in higher order ambisonics and how they play out. So we have different types of speaker arrays for doing that, including the holophonic speaker array, which kind of works on some research that was done in Canberra in Australia. Also like to build portable ambisonic systems, because I think, why can't you take this pretentious laboratory-based musical art form that's always presented in a concert hall? Why can't you just take that to the pub and do a regular pub gig? So I've been really interested in all of these projection-based systems from Institute in Graz. You might know of the icosahedron speaker that they've built at IAM. So how can you make these things more affordable and develop a culture of spatial sound? So the interesting thing about these little arrays is that if you have the right diffusion surface, that your sound is reflecting off, you get this kind of reproduction of another sound field on the wall, and you can project phantom images beyond the size of the room, and they can be very convincing. Well, they always seem to convince my husky dog that had super ears. I've been getting into spatial sound and multisensory interactions. I think this kind of musification is one way to go with sonification, but I'm really enjoying the sensification term that was first quoted by Tuck and Towett, amongst others, in the kind of the design and museum space. So I've got an augmented reality sound table that I built with my students. And the first thing I did was I put a speaker array on it. So that has like a third order ambisonic array. And what you're looking at there or not looking at is a box with sand in it that you can move around. And many of you might have seen this kind of work where you project onto it with a video from above, and you use the contours of the sand to affect some kind of projection mapping. But of course, with all of the things that I've done with sound, really, it doesn't take too long before I end up with a human in the loop, rather than just being a passive listener or studier of the data, becoming actively engaged and searching through the data somehow, or affecting some kind of interaction. So we do lots of things. Swinburne doesn't get a huge amount of government grants. We do get them. We get a lot of little grants from all sorts of other bodies, and little fish are sweet. And we also have to go out into industry and kind of, you know, as an artist and a researcher, I always feel compromised and prostituted. And goodness me, I ended up doing a PhD for an industry that was in defense. Shock. So, but anyway, it's all led to really interesting projects that we do. And of great interest is this kind of accumulation of noise in the environment and noise mapping. So it's, although these things aren't really sonification, I thought you might be interested to know what else we're doing. We're also engaged in blind and low vision projects. So dabbled in this area, and it's very difficult to get funding for this area, which is, so these things have to be passion projects, which they are. So it's part of being a musician and finding myself completely unemployable and disenfranchised from the economy. I ended up in medical research for a long time, building devices for measuring retina responses and signals. So I've always had an interest in the blind and low vision community, because I got to meet hundreds of them doing that job from all different ages, and you'd sit there doing tests with them for half an hour or an hour or so, and you've all had those tests if you're blind, and I apologize for having to do them because some of them are quite torturous. So I've always wanted to get back to that space to try and engage those people somehow, and make their lives more interesting or more easier in some way. So I've spatialized autocons in people's houses, you know, for driving their Google Books and their media and things like that. We've also done a project at Swinburne with journey planning. So a lot of low vision blind people like to hear really realistic sound immersions of places, and prior to actually going there, use that memory of the place to navigate and know where they are. We've looked at wayfinding things. We've done some reviews of the Microsoft technology, and more recently I've been working with Erica in her Monash sensory science exhibitions, and with the formation of these multi-sensory science books for STEM education, or STEAM, I should say STEAM. So onto sonification, what are we doing? Well, this is in a nutshell, and I don't want to steal Matt's thunder, so I'm not going to say too much about Matt, but we're currently this year looking at EEG sonification. So this is a really great area to get into, because there's still the physiologists that we're doing, the physicians in EEG that are now running all of these labs in Australia. They used to actually do their own sonification back in the 80s and 90s, just to speed up their review of cases. So, wow, we have tacit knowledge in space to study, and it's really, really cool. So we're looking at EEG. Our new slant to it, of course, is spatial, and there's lots of ways that we can go with that. But there's a PhD student, and Matt and I are supervising him, and hopefully he will come in and present more on that. I'm also like a closet synthesizer hack, and I've been following EEG and biomusic signals all my life. And many of these people I've met along my journey, going to things like the new interfaces for musical expression events, the International Computer Music Conference events, which I've been attending since the 90s. And so yeah, some of you might know some of these people, but if you don't, the links will be there and you can check their stuff out. So biomusic's been around for a long time, and so have synthesizers. So like I've been kind of bringing those together. This is my current modular rack, and at the heart of it is synthesizers and modules. There's a module in there called Maths. Don't ask me what it does. It's an analog computer. It's geeky. There are things in there that record and play back and loop sound and analog sequences that can go much faster and drive streams of notes faster than a MIDI interface can. And in the heart of my rig, there's a little brain interface that talks to the NeuroSky, so I can pull all of those signals out and utilize the algorithms for, alpha attention and so on in real time, as well as just getting the raw EEG data and making music out of that, which kind of sounds like this. So there's a gamut of EEG signals that are being used there. It's a live performance, and it's all done with just one monophonic voice, which is then controlled with different brain signals. And it's very, very unrewarding music to play as a musician. As having been a jazz musician and a computer musician, you're used to playing a sound or smashing down a chord or playing a sequence out of giant steps and going, yeah. But this kind of music is really different. It's sort of like, what did I just do? Was I even conscious? Was I awake? And I wanted it to go this way, but it went another way. So it's a really interesting and complex feedback loop. And I might just leave some of that music on while I talk, but just let me know if it gets annoying, because I know new age kind of music can be quite annoying. And it's not intended to be new age or meditative or anything. I'm not kind of so much into that scene. Although, some people could meditate to my music. I like to think about it as waking your mind up, being active as a listener.
I think that your background noise, the sounds are interfering with your voice great. Coming through clearly. I think it's a weird Zoom thing.
Right. Okay, I'm pulling that up. Thanks, Zoom. So, which brings me onto the multi-sensory interactions. So I've got, like I said, a bit of background in coding, and here's a coding example of the kinds of things that I do. I'll grab a set of data. In this case, there's an RNA sequence for the HIV virus. And then I try and make visualizations and things out of that. And because I'm working in a design school, it doesn't hurt to actually make something that's architectural or, of interest to my fellow colleagues in design or artistic or whatever. But it's given me a lot of techniques for grabbing data and exploring, what does and doesn't work. So you can see here that I've taken the RNA sequence, I've composed them into a number of lines of 60 elements per line, and then I'm creating color maps to those. So I guess if you start visualizing data with rainbow maps, you realize straight away that that's not going to really work. And I might actually just go to the video now. This probably explains it better than I could with slides. So I'm just going to play this.
We should explain what we do first at Monash. So these are part of the multisensory science books, are part of the Monash Sensory Science Initiative, which I started in 2018 with Professor Jamie Ross-John with his lab at the Biomedicine Discovery Institute. So I'm the legally blind artist that creates exhibitions for people who are blind, have low vision or diverse needs. And we take these exhibitions about immunology and biomedicine across Australia, and gathering world attention now with the work that we're doing, really to bring science and an understanding of science, and for people with vision impairments like me to join in the discourse around science, and really with an aim to getting people with vision impairment to say, yeah, bugger it, I'm going to have a career in science. It's irrelevant that I have a disability. And so we want to really make this kind of stuff really accessible for students and anyone of any age who wants to engage in science. To use juice, sorry.
Yeah, so you can see here in the books, there's a mixture of tactile art, and these are prototype books at the moment that you're seeing in the shop. So we've got different cells and objects and things. We're also using interaction via cameras with the React Division software that was developed for the React table. Another great Victorian developer there, Ross Benchina, who's involved in that. So you can see these funny little QWERTY code things up in the top of the corners of the books that the cameras are looking for, and they drive all the media. We did this because all of our exhibitions got shut down in COVID. Melbourne was in lockdown for longer than any other place on the planet, and it had a big effect on our exhibitions. So we made portable exhibition books that you could wash with antiviral detergents or whatever. So yeah, and I'll bring in the audio here. Actually, maybe I'll just speed a bit of this up. To be able to compose music to the gut biota, you know, around about the 1/5 of a kilogram in weight, but packing, you know, hundreds of trillions of microorganisms. I mean, how incredible is that? And how to pull that off in sound? Well, really, I had no idea in the beginning, but I've been into synthesizers since the 1970s, and I think that electronic music really has its place at creating sound for things that we really have no idea what they sound like. I mean, you could get an underwater microphone and put it down into your gut. Maybe you would hear something, but it wouldn't do anything to really depict the minutiae of the events and the amazing things that are happening between cells and proteins at that molecular level. So I've been making music, I would say. I've created about 3 hours of music for this project with a whole range of synthesizers. Modular synthesizers are fantastic for this because, would you believe, as old as they are and the technology goes way back. Yeah, sorry, I'm prattling. I just might move it on to the sonification bit here. Have their own soundtrack that depicts the art. At the same time, we've tried to turn the artwork into sound directly through a very special software program that I've been creating at Swinburne that allows you to take an image and turn it into sound. So the process of turning
data into sound is called sonification. And this process that we've adopted is a bit like a spectrograph, but in reverse.
Yeah, sorry, this is pitching at a general audience.
Spectrum and drawing a map of it. We're actually taking every pixel in a picture and then re-synthesizing that into sound. Through this technique, we're trying to create a multi-sensory experience of an image that you can feel with your hands and hear at the same time. Here are the sounds of some proteins: vitamin B, vitamin A.
Yeah, so you can see there that we're at pretty early stages of our sonification of protein data. And you know, I guess that's okay, but we've learnt a lot through that. We've learnt that so far we've learnt that technique doesn't actually really work. And the way we would like to go this year with our sonifications is to use it as an extra channel of information, and not as another way of viewing a picture, so to speak. I mean, you can do that, but I think it has limited value. It also brings artifacts into the sonification that aren't there in the original data or structure that you're looking at. So, but you can see these books are really great, and they have amazing tactile art all the way through them. What you're not seeing in this version of the book is there's also a Braille information that's in there. And Erica's maybe better to do another presentation on this for you guys. It might be of great interest, but for me, Erica brings a language that we all know, of real world tactile objects that we've grown up with, that we've eaten, that we've consumed, and she puts those into the art, and they carry scientific meaning for me. And I think that's really interesting part of this project. And it makes me feel that in some ways sonification techniques are kind of have to push into some new spaces to adjust. So that's kind of the end of what I have to say. Erica might want to say a few things, and then I know we've gone over time, so please.
No, actually, you're fine. I didn't check my email in time to see that Paul is not going to be able to make it, so we can go a bit longer here. But I'm also interested if you could give a description of the book. Go tell us the size of the book and what one would experience opening it, so that some of the audience could get a better idea of what those books really are and having them.
Yeah, sure. The multisensory science books are about A3 in size, and they're made on foam core, and then I've laminated the pages. We've got large print, we've got Braille where we can fit it, but I've got a supplementary Braille book that correlates with the text. And then we've made multisensory artworks, say of, we saw there vitamin B molecules. So we've got two topics, basically. We've got one set of books. It's 10 books on the one topic called My Goodness, which we got a grant for from National Science Week in Australia. And it's about the gut biota. So I had all the scientists from Monash Biomedicine Discovery Institute who work in that field to contribute to the books. These are world-renowned researchers working in their field. So we've got expert text, and then I've created with them, working with them and looking at what they've written and what they're discovering, created artworks that go with their particular topics. So that's one set of books called My Goodness. The other set of books is called The Heroes Within You, which is about the immune system So it's like a basic understanding of the immune system, but anyone can jump in and really appreciate the pages. So they're quite big books. They're not too heavy, but and you can see we've got the little, what do you call those again? The fiducials.
Fiducials.
Yeah, the fiducials. So you know when the page is turning, what information is going to be in there. So the thing is, it's really multisensory. We're going to have smells, and we're going to have touch, and we're going to have large text, and we're going to have braille, and the sonifications, and the music, and the text being read to you as you turn the page. So that for people, again, like me, who have got low vision, that we can actually access this material, and that we can really get an understanding of immunology and science, and really engage in it, but in a really interesting way. I really wanted to make alive the love of knowledge, and to stimulate people's like wonderment. And so these books that you can see here, they were still in production, and they're actually an evolving thing, because there are so many issues, like you said, the research challenges that underpin the making of these books, the questions that we have to ask ourselves and find answers to in how to make things accessible, and also the practicalities, like how do you fit everything onto a page with large text. And even the text that I've made there, large, I think I made 24 or 28 font, it's still too small. I still can't read it. So how big do you need to get it for someone to be able to read if they have low vision? And where can sonification and audio really support us in that? And like Stu said, it can't just be one or the other. We have to work out how to make these integrated, how they support each other, so that you get a fuller, deeper understanding of what you're looking at.
So the other thing Erica didn't mention is that there's a computer that goes with every book.
Yeah, of course.
We're using just little Apple mini computers, but the intention is to also have the software ported to Raspberry Pi for cost-effective world distribution of the books. And we've designed an acrylic kind of a bench stand as well, so for DIY people, schools, or whatever. So it's been carefully designed so we can have the books up at an angle. So we've done so far some ergonomic profiling and working out what we can do. And over Christmas we've been mucking around with the fiducials themselves, like printing them onto small pieces like drink coaster size pieces that you may place on the page to see if there's other ways to break up the linearity of each page, of the way it reads back everything. And of course, we've got those tracking on the position on the page. So, and we've got them also bringing in rotational data. So these may be great kind of widget controllers that allow people with blindness and low vision to play with the sonification. And I think for me, what's happened in my awareness of sonification in the last 6 months is that there's really an advantage to having a human in the loop. Been talking to people at Swinburne Astronomy, and they've got their sonification things going with all sorts of projects as well. They've got blind radio astronomers that are heavily involved, engaged in those projects, and they're really cool projects. They're talking about the other thing that I've been teaching in the design school at Swinburne, would you believe, is machine learning. And machine learning can only find so much in that astronomical purview. So we need that. They're talking about shared interaction between humans and machine learning. So sonification there has a really exciting role to play. And whenever a screen can't cut it, which, hey, if it's something complicated or a needle in a haystack, it's going to be like nearly every time. So, prior to this, I've been always thinking that, the sonification is really about that fabulous paper by Edwards looking at the cervical cancer slides and turning that 5-minute expert microscope assay into a 3-second brown note test that anybody can perform. And they're two like really wonderful gifts that sonification provide, you know, speeding up the process and democratizing the process to people who aren't experts. But how does it play out when it's in the sensification realm? I guess we'll find out. So we've got planned in the future some co-design. We've done co-design with the scientists so far to produce their work. And what we've learned from that is that none of them have really the communication skills that are required to go out to like a professional level exhibition. So we know that we're going to have to get editors in, or we're going to have to do that ourselves.
So yeah, there's some questions in the chat about are you one of them was along this lines of, are you collaborating with neuroscientists in the development of this, and who are your collaborators?
We have David Sly has a background in neuroscience, so he teaches in the health division at Swinburne, and he's has a strong suit though in perception and hearing, more so than say cognitive load and you know, shared interaction perception. So yeah, we're still reading and maybe looking, but if this is why it's so exciting to come to a group such as this, because there's opportunities to share, to talk, and to maybe even co-publish or co-work together.
Yeah, the neuroscience is a really good issue. How do we learn? How do we really learn and remember? How do we get stimulated and interested? How do we retain things? What are the ways in which we learn? How can we strengthen that? And you really see that with the formation of these books. And I have to say, a lot of the stuff I make, I use food and found objects. They inherently have memory encoded in them, and they have their own cellular structures as well. And they may correlate in some ways to cells of the immune system, etc. So, does that help with understanding interactions and structures at a molecular level? And there's all the philosophical questions as well as the neuroscience and science questions, the biological questions. It all converges in some delicious way on the pages of these books and the project that we're doing. So there's so many questions.
Yeah, so,
yes, Adrian wants to know if you're going to be doing workshops for educators, and how do we get hold of these?
We did something last year the year before now at Monash, making, you know, multisensory. So I remember doing a workshop with Professor Matt Butler on that. So yeah, I think definitely that's a great way to go. I think we need more. We need to get people involved in making things interactive and deliciously multisensory. And how do we do that? Well, it's fun. It's fun being a blind artist and helping people explore that, see other ways of seeing.
we did one workshop for people with blindness and low vision on like a YouTube video. And that was a challenge. How we were going to do that? So we did one for sighted people on YouTube, and we did an audio one in ASMR. Because why should all of the kind of kinky fetish people have the ASMR
why
thing?
not use it for science?
Blowing a chill wind down the back of your neck, or squirting water over an umbrella or whatever. I mean, these are fantastic ways to communicate science. So we co-opted that method pretty quickly, and that was actually really great fun. So
you can see all these on the Ross John Lab, the Monash Sensory Science tab at the Ross John Lab at Monash University. You can see that ASMR project where we called it the Couscous Project. So we had ASMR and demonstrating how to make out of chicken wire and couscous a HIV protein model. And then we've got the ASMR sounds going left here, right here, and all the delicious sounds of couscous sort of on the foil, on the aluminium tin foil, things like that. So yeah, it was really good fun. And what Stu showed before with the RNA of the HIV, we also made a 5-foot big HIV protein capsid sculpture it took me about 6 months because we used tiny little foam balls, and then Stu projected the RNA data onto this pumping HIV capsid, and we took it to the dance floor. So you can also see that on YouTube, the multisensory HIV with the projections on there. So there's so many wonderful, inspiring and delicious ways to take sonification and incorporate it into art and science.
So it would be really interesting too to if you feel free to add some of these links to the agenda from today so that we can reference that. And you also have access to the Sonification World Chat. Anyone can send out on the SWC at glasseducation.org, and that would be wonderful. And and definitely, I'm seeing your slide up now for your contact information. We would love to make sure you've got that in the agenda as well. And do we have anybody else who has any questions? And Leona put up the link to the Monash Sensory Science. So those are, appreciate those. Any other questions? Because I think what we'll do is we'll wrap up a little early today. We've just got the one presenter today. And I do want to let everyone know, I'll stick around and answer some more questions. I'm going to push the next Sonification World Chat into the beginning, to this basic same time slot, the first week in April, if that works for everybody. Otherwise, it'll have to go to the second week. But if you have an opinion on whether second week of April, I'm not going to do one in March. There are too many spring breaky things coming up here in Wisconsin. So with that, you can post in chat. Yes.
I'm just seeing a couple of things in the chat. I've got
yes.
the chat up. Sorry,
go ahead.
I didn't see questions before with the video up. But yes, our intention is to distribute the tech and the designs and everything so that people can make their own multisensory books. And that's very much our intention. But it's probably about a year away. So we have to do some research first, and we also have to thoroughly test the books with a blind and low vision focus group. So we're planning to pitch the books at the moment for kind of young adults all the way through to older adults. Like we feel that group in the community is not really getting a lot of action. But of course, there's huge potential for kids in schools and for art teachers and whatever. There's a huge chain of art and craft stores in Australia called Spotlight, and it's our plan is to reach out to them for sponsorship. And one of our dreams is to have multisensory science packs available on their website for teachers. You know, of course, the formation of all the media is quite intensive. So there's a lot of recording and collating and putting together a sonification. So our dream is to also have some kind of maybe web interface or whatever where teachers can do that and burn their own kind of audio pages. But those things are a year or two away, and we also require funding for that as well. So yeah, we're out looking for dollars and people who can commit time and be involved in the project. Yeah, but it's a really exciting project, and yeah, very happy to be involved.
Yeah, I think that would make an excellent topic for a education research grant. The multisensory approach is really amazing, and I think that connecting that as well to that ability to know what page you're turning to, and that helps so you're not floundering. That cutting down that time that it takes someone to orient to the actual tool is something I know that we have struggled with on our projects, and I think this is quite well done. And I look forward to seeing about what what actually is the mechanism by which you're doing that, so that we can borrow it.
Oh, sure. Yeah, look, I borrowed it from some old colleagues,
Okay.
the convolution brothers of, Miller Puckett and Courtlip and Zach Settle. So it's an algorithm that Zach developed. It's quite old. It's embedded in some of the Max audio software kind of latent tutorials. The keys to the castle are in there. But yeah, I'm happy to share. I'm not proud of that work yet. I think it's got a lot to do. And I've also built a physical controller to do all of those things, to get you away from the mouse, because I hate the fact that mouse driven is so engineering in its focus, and it puts you onto one parameter at a time. And really what you need to do sometimes is just tweak multiple parameters to find that juicy spot that's right in the center. That gives you a fantastic result where everything suddenly pops and becomes really audible. So yeah, I'm also working on those things as well. But I guess for me, the multisensory thing is really interesting, because it seems to be a natural extension of sonification, to kind of get it out to a much wider audience and embed it in general public displays and things. And it's one of the great things about working with Erica is that her work is a complete contrast to all of those wonderful 3D printed models that are all accurate in every scale and dimension. And you'll see this fully rendered ribbon protein in all of its complexity and stuff. And when you go to Erica's exhibition, you'll touch something and you go, oh my God, what is that? I thought it was hard plastic, but it's actually slime. And then you've got that Peter Sellers moment out of the party where you smell your hand and what is that?
It's durian.
Or it's yeast, or it's something like that. So that's another really interesting dimension as well. I've always been fascinated in olfactory display. There's, as we all are, we all dabble in collecting those because they are so bizarre, all of those projects. And at Swinburne, someone made an olfactory display that was quite sophisticated, and it was to train people to appreciate wine and judge wine. And while other people have been, looking at the effects of mood with olfactory, spraying various things through masks and stuff like that. And I think, in the time of COVID, we should also be looking at creating scents and things like that as well, and just seeing what they can do. We can dab them on a mask, or we can spray them in the air, or we can have a huge plague protecting incense burners swinging through the exhibition space complete with amazing multisensory art.
Yeah.
Who knows?
So we have these in the multisensory science book, developing little scratch and sniff things so that you can explore that on the pages as well. And how this also incorporates with the sonification of things, how they can work together. Who knows? Interesting.
Okay. All right. Well, that was really great. Really appreciate that presentation. And the reminder that the recordings will be in the folder as soon as I can get them rendered, and the chat does end up in that folder as well. So if there's something that you were looking for, it's there for you. So it looks like I have got no strong objections to the first week in April for our next World Chat. And we hope to hear from Paul Green then. And if there's anyone else who would like to present that week, let us know. And we look forward to hearing from the Learn group and anybody else who has something to share. So with that, if you have, I'm going to stay on and talk to the RCN folks for a little bit. And but if you have any last minute questions, you guys can stick around, or you're free to go and have a nice evening or morning or whatever part of the world you're at.
Can I just say that thank you so much for that presentation. It was very inspiring. but I'm also impressed that there's so many good projects here in Australia happening, and we seem to be slowly connecting. We had been in touch with Jeff Cookey and Gary Foran, etc., from Swinburne, with the astronomy, but there are so many more projects that I wasn't aware of this one. And I'm also the president of South Pacific Educators in Vision Impairment, which is the organization for all the teachers of students who are vision impaired here. So we would be interested in this project as well, especially if you're going to do workshops for teachers. So I'm going to be reaching out to you via your emails, but I just wanted to say that I think a big applause for everyone in Australia. I think it's quite amazing. What do you think, Kate? Don't you think?
Yeah, I definitely think this is you guys could, at some point you get to dominate the time zone, and we'll get it off.
Yeah, we'll pull it in 2 hours.
Yeah, I know we're getting top heavy, and so I think maybe we should try for 10 o'clock in the morning for you guys. I don't know. I can stay up a little later one time.
That's all right.
So yes, it is interesting and impressive how a lot of the really big projects are going on in Australia, and creative, diverse projects. So. And then we've got Thea's leadership, which is awesome.
Well, thank you very much, Kate. And Erica and I would love to join whenever we can. Can't